you reckon I should make a start?
Yeah, sure. Whenever you're ready.
Okay, so I've got a very good start in that I have no idea how to pronounce this because I've only read this in papers, but I'm going to be calling it APOE. If anyone knows how to pronounce it, I'm happy to recorrect myself, but... So, if I start, so APOE is a gene that codes for a polypoprotein E, and it has various different genetic variants. So it has APOE4, which this is a high risk for Alzheimer's. And if you've got one copy of the allele, you're 2 to 3 times more likely than the general population to contract Alzheimer's. If you have 2 copies of the allele, so if you're homozygous for APOE4, you're about 8 to 12 times more likely. So it's one of the biggest genetic risk factors for Alzheimer's, late onset Alzheimer's. We have APOE3, which is considered neutral risk, so it's the most common variant of this allele. Most of the population has it. You're not considered at a high or a low risk for Alzheimer's. It's just it's the normal copy of the gene. And then we've got APOE2, which is lower risk. It's a much more rare variant, and it's not very well studied because it's rare and it isn't linked to high rates of Alzheimer's. So what it is, it's a cholesterol or a lipid carrier, and I'll go on to show you the structure in a minute. So, for context, the 2 major proteins that are involved in Alzheimer's are 2 things that aren't coded for by this gene, but are contextually important to understand. So we've got beta amyloid. These kind of congregate and form plaques within the cell, and this can disrupt the cell shape. It can change the membrane potential, and it can change the shape of channel or pore proteins, which means, particularly within the brain, that can be really problematic, particularly in neurons, things like that, means it can mess with the signaling. And then we've got tau proteins, which can polymerize into these long fibrils. And I just thought it was funny that they were called neurofibrillar tangles, called NFTs. And they can, they particularly form tangles within the neurons, and then they disrupt the signaling within the neuron. Okay, so this is the structure of APOE. So here we've got, I'll come to this side and then I can point. So this is our high-risk form of protein, and this is our considered normal variant of the protein. And you can see here this, I think it's called a hinge domain, is slightly different in the 2. And this is because you've got this mutation here, R112, and it forms this relationship here which pulls the hinge protein in. And so, as you know, within a protein, if there's a mutation which changes the shape, it changes the function. And this can lead to 2 major issues. One is that it can form toxic products, so the multiglobules and the soluble aggregates, which I don't know a great deal about, but they can be neurologically toxic. And also the protein doesn't work very well as a carrier protein, and that is a whole issue unto itself, which I will go on to explain. So the first issue is its problems with the cytoskeleton transport, so there can be disruption of that. So because the shape of the protein is different, because of the way that the hinge domain is changed, it gets recognized as a dysfunctional protein and broken down. Now the issues with that is that it doesn't continue along the pathway. It's able to escape the secretion pathway and end up building up in the cytosol within the endoplasmic reticulum, within the Golgi, and then that can disrupt other protein creation, other lipid creation, and that can cause a whole host of downstream problems. And it also means, because of this changed shape, normally APOE3 is able to bind to a specific binding domain on the tau protein, which is the one that forms along the fibers. But because it isn't able to bind onto the special binding domain, it does bind to tau, but in different locations. So it forms much more oddly shaped fibrils, and so they're much more tangled, much more unstructured, and it can cause more signaling disruption within the neurons. And this is one that isn't so highly researched, the GABA dysfunction. So GABA is a neuro. Yes, go on.
Just a quick question. So APOE3 forms a complex with tau. Is that
Yes.
correct?
Yeah.
And so the dysfunctional protein ends up forming clusters that ends up forming that.
Yes. So they both form polymers with tau.
Yeah.
But the APOE3 forms a much more regular fiber, which doesn't cause much disruption,
Okay.
as far as I understand.
So with APOE4, why does that end up forming the green fiber? Is it just formed because it forms a globule in the end?
I think. That's that.
No, that's that.
As I understand, and I haven't done much further research into this, it's that it binds onto it in unexpected locations, so you have a very branched fibre, which, if that moves within the cell,
Yeah.
it can cause more disruption.
Okay.
Does that answer your question?
Yeah, makes sense.
So GABA is a neurotransmitter. I think it's called gamma aminobutyric acid, and that's a neurotransmitter involved in memory and learning. So, and how it interacts with. Actually, I don't know the function of how this APOE4 interacts with it. I don't know if it's fully understood how they interact together, or at least as far as this paper is concerned, it doesn't understand it truly. But there is a relationship between GABA and APOE4, where there's some kind of binding and deactivation of GABA, and so it isn't able to signal properly. So there is various different ways in which the mitochondria are affected by this changed protein. So there's the direct effects in that it binds to both the alpha and beta subunits of ATP synthase, which you might all have studied it, but there's the alpha and beta subunits, which turn as it creates ATP. So binding to those is going to change the way the shapes change, and it means that you are going to have good ATP production when APOE4 is bound. So that's going to reduce your ATP production, which means that your mitochondria is no longer efficient. Also, the cleaved form of APOE4, when it's these small fragments, that they are able to bind to the complexes 3 and 4 in the electron transport chain. So it means that you also don't have those properly carrying electrons. So that's also not able to produce, move the electrons properly, and it's not able to produce ATP as well either. And because of the disruption of the cytoskeleton, you also have an issue with cell transport, and that's particularly important for mitochondria because they need to be moved around a lot within the brain. So there's a lot of high energy areas within the brain that needs a lot of ATP, and so if there's disruption in neurotransport, then you're not going to be able to move the ATP to areas you need, particularly when new synapses are being formed, new pathways are being formed, you're not having proper mitochondrial distribution. So, there is one gene relationship which is still being studied, and they're still not quite sure exactly how the relationship between these two work. But APOE4 and all the APOE and TOM40 are both on chromosome 9. They're reasonably close to each other, and they have a linkage disequilibrium, which means that they are somehow associated with each other while not being in the same location. And the presence of APOE4 ends up downregulating production of TOM40, which is this pore-forming, a particular component. I think that's there, this pore on the outer membrane of mitochondria, which means that you have issues transporting proteins that are working within the mitochondria, and that again means there's lower efficiency, particularly because you need, I think 99% of the proteins that are used within the mitochondria are imported, despite there being genetic material inside. Now, the reason this is interesting, APOE4 to study, as opposed to the other proteins, which are also being heavily studied, is there is actually something that can be done about the effects of this. So there is these small molecule correctors. I think there's various different ones that are being studied currently, and I don't think they've got onto human trials yet. They'll testing more mice. And they're able to change the structure of the APOE4 so that it's shape-wise quite similar to APOE3, and that means it's no longer recognized as a dysfunctional shape, and that means there's no cleavage, and that means the binding happens much more correctly. So there is research being done into whether we can use that to undo some of these extra effects that lead to much worse symptoms of Alzheimer's.
So do you know how it actually corrects it? Is it just acid substitution, or is it changing something else?
As I understand, and I want to research this more, but I've only had 2 days of the paper, so it manages to disrupt the relationship between these two, what was it?
Arginine
and glutamic acid, and it enables them to be spaced out. I'm not quite exactly sure how it works, but I imagine it interacts with amino acids either side.
Yeah, so would it be potentially dangerous depending on the mechanism in which it disrupts the interaction? Because,
yes,
you know...
well that's why it's still undergoing trials.
Yeah.
So it hasn't killed off the mice that it's been tested on so far, but there's still a lot more testing to see about safety and whether or not it's a system that can be used on people.
That's what I'm trying to figure out. I mean, it has to be, it's a certain size, right? So I'm assuming it's forms some sort of like molecular bond in the middle and it like forces things apart.
Yeah.
So there are only so many proteins with that kind of structure, I think.
Yeah,
Because
they're very
this is
small. I was looking at
on
pictures
there.
of them, and I think it's only a couple of amino acids long. It's really tiny and wonderful. And I think there's a lot of different ones that they're still trialing. So there's various different ones that do work to correct the shape, but they're still under progress. Right, I think that's all I've got to say about it. Is there any more questions? I feel like I've raced through that.
Yeah.
Sorry, can I ask,
Yeah.
how is this APOE4, like the protein that's supposed to adjust the APOE4 shape? How is it like expressed? Or like how is
I
it
don't
delivered
know.
into
I don't even know if they've begun inserting. Well, I don't know much about this because the article I was reading, and I did do some additional research, was just positing it as a future advancement that might be made. So it hasn't had a great deal of research done into it yet.
Yeah, so do we know like this mutation occurs after or like before
Which
birth?
one? This mutation here?
Yeah, this should be like predisposed, right?
Yes. So it's familial, and that is partly how familial Alzheimer's gets passed
So how does it like manifest at such late age? If
I don't know.
so, it just aggregates for like 50 years in your brain.
No, I thought it was in the paper said something about methylation. How you have different amounts of methylation
okay.
and how that's
that
constantly
might be what it is.
regulated over
Yeah.
time. But as you get older, that methylation process doesn't keep going on. So then it ends up being
That makes sense.
Or would be something in the microbiome?
Yeah, it could be microbiome.
But it's quite rough if both the parents are carriers and then you end up homozygous for it, you have a much, much higher chance of the onset of Alzheimer's, which I think is about 65.
So this methylation thing, is it possibly an evolutionary mechanism to counter this APOE4 effect until you get older? Or is it just...
I'm not sure, I didn't. I only
But you
looked
can
at it
imagine
for
that's a
5 minutes.
selection pressure,
I think it's very
right?
long.
I mean, it makes sense as a selection pressure, right? Because
Yeah.
particularly with late stage Alzheimer's, you aren't really able to care for yourself, so.
Because at that point, you know, genetic material has already been passed on, so it's
Yeah.
like evolution doesn't care.
Yeah,
anymore.
yeah.
It's like, maybe they just like, you know, the methylation lasts as long as
Yeah.
you know, you pass the. Just
Right. So...
I think also, I know I did a little bit on Alzheimer's a while ago, which wasn't related to
Oh, yeah,
people, but thinking about like as it manifests itself, it's also because the aggregates build up as the cerebral spinal fluid isn't cleared as well if you're APOE4 for some molecular reason. So that also could be why it only manifests later on, because it's just a build up over many years.
Well, I was researching about this yesterday, and there has been a very recent update. So what they've been researching a lot about for the last 20 years is beta amyloid accumulation and within the brain and then forming these big plaques, and they thought that might be the route to a cure. So there's been loads of different trials of various different drugs that are able to break down the plaques. None of them have worked. And recently, one of the quite pivotal papers that came out talking about a specific kind of beta amyloid that was involved turned out that the images within it, so I think it's a Western blot, has been forged. And so there's all sorts of controversy around that recently. And a particular scientist that was involved with this area of research has come under a lot of scrutiny. A lot of his research is under, all of it's being reinvestigated. And there seems to be a lot of questions around his results that show, and no one was able to replicate his results for this specific kind of beta amyloid. So, yeah, it's definitely not looking good.
If we don't have any other questions, we can move on. Yeah, so thank you, Briony, for bringing us this presentation. And would you like to go? Right, so anyway, this paper is about like how we can kind of like restore kind of like the methylation age, like we can reset the clock by like certain transcription factors, and we will also do some kind of like demethylation with it. These are like just a bunch of like Harvard researchers, and they're like, this paper is like on Nature or something, so I just found this paper and I'm like kind of interested in it. Anyway, so just some background, you might probably know some of it. Aging is just like the overall degeneration of an organism, and basically it's like the buildup of epigenetic noise will contribute to it. And one of that includes like the DNA methylation patterns. So basically this will like impair our tissue function and also our regenerative capacities. And so scientists do have like this question whether we can like restore aging by restoring some kind of like a sort of youthful methylation pattern. So to do that, we have to have some kind of blueprint of like our original like young methylation pattern. And they would like also want to do, like kind of investigate whether this blueprint exists, right? Yeah, so these are just like some previous studies done in the area. Some studies about like the Yamanaka factors, we're going to go into that later. Basically, they're like a group of transcription factors, and they're like responsible for like reprogramming cells, and they can also restore the like the pluripotency, not exactly pluripotency, but like multipotency of like the stem cells. However, there is this issue that the continuous expression of all these four factors in previous experiments did contribute to like oncogenesis, which we do not want for any stem cell therapies. So this attempt is to see whether a controlled expression of three of these factors, as we'll go into later, will actually contribute to like reversing the aging of like especially the CNS, the retinal tissue, while like not causing cancer. So yeah. So at a glance, this study is about like how like kind of like an externally induced expression of three of these factors. So we've talked about OAT4, SOX2, KLF4, and MYC, right? So we would just abbreviate these as OSKM, and in this study, the M factor, like the MYC factor, is excluded for reasons we'll go into later. And then they also need to like rely on the action of the demethylases to demethylate the DNA and basically restore the methylation profile. And then this is done in mouse like retinal ganglion cells. Basically, they use the eye as a model of the CNS. So they kind of like postulate that if this is successful in the eyes, then it might be a promising solution for like CNS regeneration, like neuroregeneration, etc. Yeah. Basically, okay, first of all, we need to know why they excluded this M factor from the study. is excluded from the study because previous study shows that this M factor, when expressed, it actually reduces the lifespan of the organism despite reprogramming cells, right? So it does induce some kind of like a stemness into cells, but it just reduces the lifespan of the organism as a whole. So therefore, if you're like kind of like using this for stem cell therapy to like counter the effects of aging, you will not want your lifespan to be reduced. So anyway, there's this 5-day expression of OSK in old mice fibroblasts and to monitor the expression of aging-related genes. So after this 5-day expression, they kind of like find a youthful mRNA profile. Youthful as in like it matches more closely with some kind of like 1-month-old mice. So old mice would basically be like 11 months old. That would be like considered old for mice. And there is like no loss of like a cellular identity or no oncogenesis either. This gamma expression is just like kind of oncogenesis, I think. Yeah. And then the 4 genes are, it proves that like OSKM is like the 4 of them. These factors are adequate for like reprogramming the cells. But yeah.
So they used the 5-day expression, so the fibroblasts, those were in vitro, and they compared it with an mRNA expression from in vivo mice?
Oh, so basically they extracted like the mice, like the young mice cells, and then do like the mRNA profile.
So it's like an ex vivo.
Yeah.
Okay.
But like their end goal is to like test whether this works in vivo because we want to like administer it to humans, so we'll get onto that later. So they then need to like test after. So the first thing they did was to test whether they can exclude NYC, and they proved that they can because OSK alone can reprogram the cells. Now we need to see whether OSK expression in vivo is actually safe for us. So basically they used this adenovirus thing to kind of like induce the expression of OSK in the mice. And they did use some kind of control on the promoter, like the recombinant promoter they put into the virus genome. So this control basically consists of 2 elements. We will talk briefly about that later. That's TET on and TET off. TET on is like when this DOX is present, you would need our TTA to like kind of like co-express this promoter. And then the TET off like activation mechanism is when DOX is not present, you would need TTA to kind of let this promoter be activated. Yeah. And then this doesn't matter. Yeah. So then they want to like co-infect the young and old mice. You can see young mice here is like around 5 months old, and aged mice is 20 months old. I'm actually not sure about like there's an inconsistency in the paper. I've seen like in the later experiments they used 11 months old as like old mice, so I'm not sure why they use like different ages of mice for different experiments, which should mean the same thing. I don't know, maybe 20-month-old mice died or something. Anyway, the OSK expression was induced by putting dogs into their drinking water, and then this will like, as we can see, lead to a TET-on mechanism, like the TET-on activation mechanism with the dogs present. So basically, they see that there's like no increase in the rate of tumorigenesis or any other negative effects in terms of the cell function. Remember, this is done in vivo, right? So it's just injected into the mice. And then you can also see like the cellular identity is maintained in the OSK expressing cells, so no oncogenesis, basically the same thing. Yeah, so and then they would like to see, after proving that in vivo injection is safe, they would like to see if it actually reaches like the goal to like stimulate axon regeneration. So what they did is they kind of like crushed the optic nerve of the mice, and then they kind of like induced OSK expression by this TET-off promoter activation. So this time there's like no dots. And then they basically injected this virus carrying the OSK polycystron. So remember, O, S, and K are like 3 different genes. So that's like polycystron with like 3 of them. And then they just injected it into the vitreous body. The vitreous body is like the liquid between the lens and the retina. And then what happened was that there is like gene expression in around 40% of the RGCs. RGCs are just like the cells, like the retinal ganglion cells that we are administering our studies on. And we can see that this continuous expression, once again, it did not induce any type of tumorigenesis, but it does kind of like restore the axon, as we can see later. So to quantify axon regeneration, they tagged this cholera toxin subunit B, which binds to our axon, and they tagged it with kind of like a fluorescent protein. And basically what happens is that the most axon regeneration and the cell survival occurred when all of OSK were delivered within the same virus. That means it's like a polycystron with all the genes. But when they're like alone, or when it's like an OS dual cystron, the effect is like minimal. So it proves that you have to like have all of them in a polycystron to like administer that kind of regenerative effect. And this can be in part explained by the fact that if you separate them into like the genomes of different AAVs, the co-infection frequency tends to be quite low. So basically, if you don't have like all 3 of them at the same time, the effect is like not significant. And then basically after 12 to 16 weeks, we can see like new axons, regenerated axons, they grow in length, and then this extends into the chiasm, which is the kind of like the interface between the axons of the ganglion cells and kind of like the CNS. And anyway, the suppression by DOX also prevented the regenerative and survival effects. So this is, again, suppression by DOX treatment means that because it doesn't have like the RTTA, which is like required for the TET-on activation, if you induce DOX without that kind of like co-activator, the expression is prevented. So you can see that if the expression of OSK is prevented, the regenerative and survival effects are gone. Yeah, so we do have a kind of like a problem here because they use, as you can see, the inconsistency here. They use like cells from like 12-month-old mice, and it works. But then in like slightly older mice, I think in the paper is around 18 months. There's like a weaker axon regeneration, and then I think there's like no increase in RGC numbers, so there's like no additional proliferation. So basically what it does is like, basically it just regenerates the old stuff, the lost stuff, and it doesn't generate like new cells. Yeah. And then like the OSK was acting directly on the RGCs rather than through the amacrine cells. They need to like prove this. Because the amacrine cells are just like neighboring cells next to the RGCs in our eyes. So basically, as we don't have much time here, we'll just see that the expression of a certain gene, a marker gene for like the regeneration, is not mediated by like that expression was like not found to be increased in the amacrine cells, but it's only in RGCs. Right. So we also need to like explain why OSK can counter the methylation aging. Basically, previously we did mention that OSK can stimulate axon regeneration. But then you can see like here, it doesn't prove that regeneration, like it works on regeneration, but can it reverse the aging profile? So now basically, what they did was to like co-express this with the methylases we talked about earlier, like the TET1 and TET2 demethylases. and this will like demethylase the DNA. So OSK will kind of like induce their action. And you can see like the untreated RGC's aging was accelerated, but then with the OSK treatment, that kind of like aging expression, like the expression of like aging mRNAs, were like counteracted. So this means that OSK can kind of like counter aging. And why did I say injury-induced aging? It's actually because they found like the injury-induced, like after post-injury, the DNA methylation profile was kind of like reminiscent of that of the aging mice profile. I don't know why that's the case. Maybe it's because there are similar functional laws, but. And then regeneration requires DNA methylation. This is basically another test which proves that demethylation is like the main thing that happens for this kind of like OSK-induced regeneration to work. So it's like not about other factors. And then basically, finally they tested like how axons regenerated in humans. So basically they did kind of like the same thing. They induced kind of like aging by the administration of vincristine, and this vincristine will induce axon injury in without like any cell proliferation. So basically it's also like injuring the axons and then by this chemical, and then they say that this is inducing aging. And then like 9 days after this so-called injury, that means like the vincristine injection, we can see like there is like a regenerated neurite area. So you can prove that there's like regeneration and the proliferation of new cells, not to the extent of oncogenesis, but there's like new cells. And you can also see like the regeneration is blocked by this knockdown. This has to be like explained by the previous stuff. Yeah, I'm not going to that. Anyway, basically the final thing is that they tested whether this can help the mice with glaucoma to recover from their vision loss. So glaucoma is just like the loss of RGCs progressively, and this is often due to increased pressure in the eyes. So basically they just inject some microbeads into the mice's eyes, and then they sort of like inject those AAVs with the OSK polycystron after 4 weeks, and that leads to like a significant decrease in the number of RGCs. So that means it's just like proving that there's an injury caused. And then afterwards, they induced OSK expression in the mice for 4 weeks, and then they see that the vision kind of equity is restored in those mice. So they use this optomotor response assay, which I think is like testing the neuron sensitivity. I'm actually not sure that's like some medical sense of. Yeah. So in conclusion, it's seen that it's possible to safely reverse aging of CNS and other tissues to restore its biological function in vivo. That can be done by inducing the expression of OSK, and this has to be done in week of like the demethylases, as we've said before. And we can also see that it's safe because there's like no observed oncogenesis, and OSK would promote a youthful gene expression. So we can see that epigenetic reprogramming could indeed promote tissue repair. However, the author did include some final thoughts on this topic. Firstly, DNA methylation might not be the only epigenetic marker involved in the reprogramming procedure, because transcription factors talked out about elsewhere in the paper, which we didn't go into details. They're also they're involved in maintaining the cell stemness, and they're associated with signature sites as in like the sites whose expression would kind of like be correlated with the organism's aging. And the cells encode and store youthful genetic material information, but we do not have like like concrete evidence on how they do it. The author suggested some kind of DNA modifications or binding proteins, but then we do not know how it actually happened. Yeah. So this is a lot of information, a lot of that which I skipped, and if you have like any questions, I can like try to answer. Do you want to like revisit any slides?
I was just curious about like the tet on, tet off, because I don't think I exactly understood.
Okay. Basically, tet on, tet off is like two mechanisms by which like the promoter, like that promoter is actually the promoter of that polycystron. The polycystron is like the OSK factors. So basically, they put the promoter and the OSK polycystron into the AAV genome, and then they use the AAV to inject the cells, infect the cells, and then the cells have the polycystron. Basically, tet on is like when the tetracycline derivative, DOX, is present, it will require this transactivator RTTA to activate the promoter.
So is DOX like within the cells that were infected?
So it's added into the drainage. So
Yeah.
I should say over
Oh,
time.
okay.
Yeah, so you can see like they kind of like, I don't know, yeah, was added into the water or something.
It's pretty smart.
So did they test the long-term effects after the rescue? Because you said at the glaucoma they rescued that. Did they say anything about the after effects or?
You mean like after the vision was restored?
Yes. Was it maintained? Or
No.
Yeah.
Yeah, they didn't say that. They just said that like the vision was restored, and then they said that there's no oncogenesis, no observed negative effects. But obviously we don't know the aftermath of the mouse who apparently, I don't know, recovered from glaucoma. So yeah, it still requires like additional testing. They did test it on human ganglion cells, and it did promote axon regeneration. But once again, you don't really know the long-term effects apart from the fact that it apparently doesn't cause oncogenesis.
Can I ask how do we test whether like the mice will develop some tumors somewhere, and how like sensitive is this test?
So you can do it in several ways, but well, there are tumors that you can see, and tumors that you can kind of like test by testing the kind of like gene expression profiles. There are several oncogenes that will be like highly expressed when that cell is like cancerous. So yeah.
There's probably a microarray that you can use to test for different.
Like you have like probes on the microarray, and then they kind of like, or can you use RT-PCR?
Yeah, you could. But you just need, it's just basically a screen for a bunch of things.
And I'm actually like not sure about this ribosomal DNA thing. Are there like any other ways you can test about the methylation age? Because the paper justifies like using ribosomal RNA by, like DNA by, because it's highly correlated with age and it's got like good site coverage of like all the methylation sites. I'm not sure. Are there any other ways to test age?
There are methylation profiles. Like
Yeah.
you can like just look at, how many methyl tags are on DNA. But the problem is there's also all this stuff going on with histones, right? Which is much more difficult to pick up.
So they did say it's.
Yeah, it's so true.
So if you guys don't have any questions at the moment, I can send the PowerPoint to you guys if you want. And
So wait, sorry, did you say that it didn't really work for the older mice?
yeah. So
Just the one
what
that
it
they
meant
really
by
wanted it
like.
to work for?
Just go back to the thing. Oh yeah, here. Right, so what it meant was that it required slightly longer treatment. So for the other mice, they need like 2 weeks, and then the axon's already back and working. However, for the slightly older mice, I think it's around like 18 months or something. I'm not sure, I have to like check the paper, but it required like more treatment. And that just means like more cost for the treatment if you're like an elderly patient and you're a human and you're glaucoma. And then there's also like no increase in like the cell numbers. So basically what it means is that the axon has regenerated back to how it should be, but there's like no new cells. So you can see that this does not solve like the intraocular pressure issue, like this kind of like a regeneration. So basically that means after the treatment, you still have like that intraocular pressure issue going on. And what will happen is that the RGCs will continue to die, and then after a while you have to like go back to the clinic and have this OSK treatment again, which takes more money than you would if you're like younger, because it takes longer.
So is that saying it's just treating the symptoms, it's not treating the cause?
Yeah, you can say that, because the cause is actually partly because of like the intraocular pressure, and this can be due to blood pressure or other issues. And then the paper also mentioned that it can treat like the loss of vision due to aging, and I think that would be like more beneficial to like the patients, because that can be like reversed without it occurring again. If it's glaucoma and you're constantly the RGCs are constantly under pressure, then if you just regenerate the axon, it will like just come back after a while. Yeah. And are there any other questions? If not, we can like pass on to the. Yeah, right.